stem cell therapies. And I will also be talking a little bit about signaling technology and how signaling technology can help stem cell therapies. Now, there are different sources of stem cells, and stem cells, the embryonic stem cells, are obtained from the 8-ball basilar stage of mammal embryos. And there have been studies on murines indicate that these cells display a tendency of developing tumors and teratomas after a few divisions. So they're very unstable. They multiply quite well, but they're very, very unstable. This is some of the embryonic stem cells in a picture here. So they are desirable, but there are problems with them. Now, stem cells can be used to treat a number of illnesses and have treated a number of illnesses, and they have even been used to deliver cancer, anti-cancer drugs, directly to breast cancers. This is a study that was done in the Kansas State University in 2008, and the delivery was done through nanoparticles of stem cells. Now, the umbilical cord stem cells, it comes from placental blood in the cord of the human umbilical cord matrix, are very stem cell rich, and they are also loaded with nanoparticles containing doxorubicin, a widely used chemotherapeutic drug. And this is one of the techniques used to treat breast cancer that has been quite successful in the United States. Now, another type of stem cells is the adult stem cells. The adult stem cells, or for short ASC, are harvested from bone marrow. That is a very painful procedure, and it doesn't get too many stem cells. It has a low yield, and it has a high morbidity, so you're losing quite a lot of stem cells in the process. A peripheral blood, the HSC, the stem cells from the peripheral blood, is another source of stem cells. Diseases in more advanced stages seem to better respond to blood stem cell transplants because obviously the blood is carrying antibodies and nutrients to the cells and oxygen, which are absolutely crucial for a lot of illnesses and obviously for the survival of the organism. The third source of adult stem cells is the fat, and we call them MSCs or ADSCs. They have a low morbidity, in other words, they last longer, and it comes through liposuction, which we have a lot of those stem cells, and of course we have a very high yield as a result of that. We have about 5,000 CFUs per gram of extracted fat. So it's quite a lot of stem cells come from the fat. And this is probably the main source of adult stem cells used today. This is a microscopic view of a colony of undifferentiated human embryonic stem cells. And the state of the art, how do we get stem cells from adipose fat? First, we have an abstention of human adipose tissue, either through liposuction or lipectomy. Then the adipose tissue is placed into sterile, ice-filled plastic bags, which are then sent to the laboratory. The adipose tissue is septically homogenized and repeatedly washed with BBS inside a biological exposure chamber. And there is centrifugation and isolation of the SPF. Interruption of the digestion after 1 hour with the addition of function of VPBS. This is a factor that basically is a treatment factor to get to the next stage. The fat and the bones